Now, continuing on our genetics journey, I wanted to just make sure that we understand each other and that we understand very important terms here. So, when we're looking at the chromosomes inside the cells, remember that for one thing, that the chromosomes are only visible during mitosis, right? Chromosomes are only a compaction level of DNA, and they're compacted in such a way that they're easy to divide in each daughter cells after mitosis, right? So during mitosis, so that after mitosis we get two daughter cells. That's what I meant to say. Now, when we're looking at every single cell before it gets to divide, actually when it's probably in phase G1 of the cell cycle, so where most of the cells will spend their time, most cells will have two copies of each single chromosome. You might not see them because they're not compacted correctly or accordingly at that time, but the chromosomes are still there. So we know that for every single cells, we will have two chromosomes inside that cell. And we know that for human beings, we have 23 pairs of chromosomes. So that is two chromosomes number one, and then we have two chromosomes number two and two chromosomes number three, etc., etc. So for a total of 46 chromosomes for every single human cells. Okay, that doesn't apply to flowers, doesn't apply to other kinds of animals. 23 pairs is a human thing, right? Some other animals have more, or can have more than 46 chromosomes per cell. Some other animals might have less. Some organisms have way more DNA than we do. Some have less. Okay, so but let's just keep it simple for now. So for every single cell, we have for this, so let's say that this is chromosome 1. Okay, let's say chromosome 1. We have two copies of the same chromosome, and we call those chromosomes, we call them homologous chromosomes. Okay, so for every single cell that has two number 1 chromosomes, well, those are homologous chromosomes. The two pairs of chromosome 2 are homologous chromosomes as well. Now remember that when the cell is just about to replicate, just about to divide, remember that in the S phase of that cell, right, when we get to the S phase, the DNA will double, right? So we have a certain number of chromosomes. We have 46, and then from 46 we will move very temporarily to 92 chromosomes before the cells divide again into two daughter cells, that each of them having 46 chromosomes in each. Okay, so during replication, each chromosome here is going to be replicated in two, and those are called, this one and this one here, they are called sister chromatids. Okay, sister chromatids is seen only when the cell is about to divide, and then when the cell divides again, right, we end up with two homologous chromosomes again. We see that those sister chromosomes, right, not chromatids here, sister chromosomes here, are now in separate cells, and those again, if we are talking about chromosome number 1 and chromosome number 1 here, we end up with two homologous chromosomes in each of these cells. So please remember that, because that's important for the rest of this presentation. Now, one thing that I want to say as well is this. When we look at the cells, and we know that cells have pairs of chromosomes, right? So we say that if there's two chromosome number 1 and number 1, number 2 and number 2, right? So we have all of these chromosomes. We say that in this case, because there are two copies, we say that the cells are diploid. Okay, diploid. So it means that there's two copies of each chromosome. However, when the cell is fertilized, okay, so when the cell is fertilized, so we have a sperm cell, and that sperm cell is going to fertilize an egg. This sperm cell here is actually haploid. haploid. And this cell here, the egg, is also haploid. So when the sperm cell fertilizes the egg, there's two haploid cells, which means that they only have one copy of each chromosome. When they merge together, they will merge into a diploid, right? Because they bring, each of them, they bring their own chromosome, and they will form a diploid fertilized cell. And that cell is known as a zygote. Okay? A zygote is a cell that was fertilized. So after fertilization of the sperm cell with the egg, both of them being haploid, bringing with themselves only one copy of each chromosome, each of them, these cells, when they merge together, they will form a zygote. Now remember that, because we will talk about heterozygotes, homozygotes, and all of that in the next few slides. So that's an important term for you to remember at this point. So what we can see in this slide is two human beings, and we have homologous chromosomes found right here. So we have chromosome here and chromosome here. You know, let's say that this is chromosome number, 16 or something. It doesn't really matter, right? So we have two homologous chromosomes, and don't forget that one of these homologous chromosomes is inherited from your mother, and the other one is inherited from your father, right? So those two cells, when they merge together, the sperm cell from your father and the egg from your mother, when they merge together and form a zygote that eventually became you, right? They brought each one, brought one copy of their chromosome number 16, right? So we decided these were chromosome number 16. It doesn't matter. It could be any other chromosome. Now, remember that when we looked at the concept of the gene, right, we know that the gene is a piece of DNA that's found on your double helix, right? When we look at the double helix like this, well, a gene is a portion of that double helix right here, and that gene is going to lead to the production of a protein, right? Will lead to the production of a protein, and these proteins are the workers of the cells. They are the ones that do all the work. Those genes are found on specific sites on the chromosomes, right? Specific sites on the chromosomes. And of course, if we're talking about chromosome 16 right here, we know that on chromosome 16, there's many parts of these chromosomes where we found very important genes, right? And when we look at some of those genes right here, we know that there are two copies of those genes. There's one copy right here in white and another copy here in blue. And it's the same thing. It's the same thing in males and females. It doesn't matter. We have here two copies of the same gene on that same chromosome, chromosome 16. One inherited from the mother, the other one inherited from the father. We say that for this one gene here, I don't know what gene that is, right? But from what we see here, we see that there's two copies of that genes. And then one very important term to remember here is that those genes are known to be alleles. Okay? What it means is that an allele is a copy of a gene. An allele is a copy of a gene. So we have, for each gene inside of us, we have two alleles. And that's an important part of that. Okay, let's move on. So we've seen that for this copy of these two chromosomes here, we decided those were chromosome 16. We know they're homologous chromosomes, and we know that we have two alleles here. Now, if you notice, when you compare these chromosomes here to these chromosomes right here, the alleles from these chromosomes here are different than the alleles from these two here. You see that there are two different alleles depending on what chromosome we are situated, right? So we have one allele here, and that allele here, right here, this one, is of course different from this one here, and we know that because of the different color. Now, on that side here, we know that those two alleles situated on the same chromosome, 16, are two of the same alleles, right? Blue and blue. So there's a name for that. The name for when we have two alleles are the same, we say that a person that has two alleles that are, that are different here, we say in this case that this person would be heterozygous for that allele, right? So they might have an allele, for example, that codes for blue eyes and the other one that codes for brown eyes, and the result of that is the effect would be a brown eye phenotype, for example, right? So on the other side, right here, we have the same alleles, and in that case, we say that this is, of course, homozygous. So if you are homozygous for blue eyes, so which means that both of your genes, both of the alleles inside your cells are coding for blue eyes, and your phenotype is blue eyes. So basically your genotype says, well, blue eyes and blue eyes, two chromosomes that have the variant of blue eyes, and the phenotype will be that you do have blue eyes. Now, if you are heterozygous, well, that can mean maybe another story, right? Where you might have one of the alleles is blue eyes, but you might have brown eyes anyway, because you have another allele that says you're going to have brown eyes. Now, we're going to explore these things a little bit further down the road, but I just wanted to mention this. So what's important here is to remember the terms alleles. It's important to remember that what is an heterozygous chromosome or person, or homozygous, right? So it's important to understand that and being able to play with those terms. Now let's say that the two models we had in the previous slide have children. So we know that we have an heterozygous right here. We have homozygous right there. So the genotype here are the two genotypes are different, and of course the phenotypes are probably going to be different as well. And we know that those two people, if they do have children, they might pass on their genotype to their children, right? So there'll be different combination that will be inherited down the road, after the next generation, right? So this here, we say that this here is F0. So these two people decide to have children. They do have children. You have a generation of children, and then what we have here is F1. Okay, F1. Now we will look at this a little bit more in details, but I just need to introduce you to someone that made a lot of advances in early genetics, and of course that person